Hello, future scientists! Today we are going to unlock one of the most fascinating processes happening inside our bodies and in the world of living things. This process is called meiosis. Think of it as a special kind of cell division. It's the reason why you have a mix of traits from your mother and your father. Meiosis is essential for sexual reproduction, which is how most animals and plants create new life. The main goal of meiosis is to create special cells called gametes. In humans, these are the sperm in males and the egg in females. These gametes are unique because they have exactly half the number of chromosomes as a normal body cell. This is super important for keeping the chromosome number consistent from one generation to the next. Imagine your body cells are like a library with 46 books, organized into 23 pairs. Meiosis is the clever librarian that carefully selects one book from each of the 23 pairs, creating a new library with just 23 books. This ensures that when the sperm, with 23 chromosomes, and the egg, with 23 chromosomes, meet, the resulting baby, or zygote, will have the correct total of 46 chromosomes, just like its parents. Just like there are different flavors of ice cream, there are different types of meiosis, depending on when and where it happens in an organism's life cycle. The first and most common type in the animal kingdom, including us humans, is called gametic meiosis. It is also known as terminal meiosis because it happens at the very end of the process, right before gametes are formed. In this type, a diploid organism, like an adult human, has specialized diploid cells, the myocytes we talked about. The second type is called zygotic meiosis or initial meiosis. This type is the complete opposite of gametic meiosis and is mostly seen in simpler organisms like fungi and some algae. In this life cycle, the main organism is actually haploid for most of its life. Two haploid gametes fuse to form a diploid zygote. Instead, the zygote itself, being the only diploid cell in the entire life cycle, immediately undergoes meiosis. The third and final type is known as sporic meiosis, or intermediate meiosis, because it happens somewhere in the middle of the life cycle. This is the strategy used by all land plants. These organisms have a fascinating life cycle called alternation of generations, where they alternate between a diploid phase and a haploid phase. The diploid adult plant, called a sporophyte, undergoes sporic meiosis. To summarize these three types simply, think about when meiosis occurs. In gametic meiosis, which happens in animals, it happens to produce gametes. In zygotic meiosis, which happens in fungi, it happens right after the zygote is formed. In sporic meiosis, which happens in plants, it happens in the middle to produce spores. Before we dive deep into the specific steps of meiosis, let's talk about another, much simpler form of cell division called amitosis. Amitosis is also known as direct cell division. The name itself gives us a clue. A means no, and mitos means thread, referring to the chromosome threads we see in meiosis or mitosis. So, amitosis is a division without the fancy, organized steps of forming chromosomes and spindle fibers. The process of amitosis is remarkably simple. First, the nucleus of the cell starts to get longer and develops a constriction, or a pinch, in the middle. The nucleus continues to stretch and eventually splits into two, without any of the chromosomes becoming visible. However, there's a catch. This division of the nucleus and its genetic material is often unequal, so one daughter cell might get more genetic material than the other. For this reason, amitosis is not the primary way healthy cells in complex organisms like humans divide. Due to its simplicity and speed, amitosis is found in certain specific situations. It can be seen in some lower organisms like bacteria and yeast, where rapid multiplication is key to survival. In more complex animals, it's usually found in cells that are highly specialized or are not in the best health. Meiosis is not a single event, but a two-part play. The first major part is called Meiosis I. This is where, honestly, the most dramatic and important events happen. The main goal of Meiosis I is to separate the homologous chromosome pairs. This first division is the real reductional division because it reduces the chromosome number from diploid, which is 2n, to haploid, which is n. The first and longest phase is prophase I. The chromosomes start to condense and become thick and visible. Then, the homologous chromosomes find each other and pair up, a process called synapsis. This pairing forms a structure called a bivalent or a tetrad. Now comes the most magical part crossing over. the paired chromosomes get so close that they can exchange small segments of their DNA. This genetic shuffling creates new combinations of genes on each chromosome, which is a major source of genetic variation. Next up is metaphase 1. During this phase, the paired homologous chromosomes, or the tetrads, line up along the middle of the cell. This middle line is called the metaphase plate. The way these pairs line up is completely random. This random orientation, known as independent assortment, is another huge source of genetic variation. Following metaphase 1 is anaphase 1. The spindle fibers start to shorten, pulling the homologous chromosomes apart from each other. It's very important to note that the sister chromatids, which are the two identical halves of a single chromosome, are not separated yet. Finally, telophase 1 completes the first division, and cytokinesis divides the cytoplasm, resulting in two haploid cells, each with half the original chromosome number. After the first division, the cell takes a very brief rest, and then the second act, Meiosis II, begins. This second division is much simpler and faster than Meiosis I. The main goal of Meiosis II is to separate the sister chromatids. Prophase II quickly prepares the chromosomes. In metaphase 2, single chromosomes line up individually along the middle of the cell. Then comes anaphase 2, the moment of separation. The centromeres holding the sister chromatids together finally divide, and the spindle fibers pull the now separated sister chromatids to opposite poles. Finally, telophase 2 and cytokinesis complete the process. The final result of this entire two-act performance is 4 haploid daughter cells. Each of these 4 cells is genetically unique, carrying a new mix of genes, ready to play its role in reproduction. So, after journeying through all these phases and types, let's zoom out and appreciate the grand importance of meiosis. Meiosis is not just a series of complicated steps happening inside a tiny cell. It is the fundamental engine of life's diversity and continuity. By halving the chromosome number, meiosis guarantees that when 2 gametes fuse during fertilization, the resulting offspring will have the correct stable number of chromosomes. Without this clever reduction, chromosome numbers would double with every generation, leading to chaos and the end of a species as we know it. Beyond just maintaining chromosome numbers, meiosis is nature's greatest artist, constantly creating new genetic masterpieces. The 2 key mechanisms for this creativity are crossing over in prophase I and the independent assortment of chromosomes in metaphase 1. This incredible genetic variation is not just for looks. It is the raw material for evolution and the key to a species' survival. A population with a wide variety of traits is much better equipped to handle challenges like changing environments, new diseases, or predators. If all individuals were genetically identical, a single new disease could wipe out the entire population. But because of the variation produced by meiosis, some individuals will likely have traits that allow them to survive and reproduce, passing those advantageous genes to the next generation. In conclusion, the phases of meiosis might seem complex, but they are part of an elegant and precise dance that life has perfected over eons. From separating homologous pairs in Meiosis I to splitting sister chromatids in Meiosis II, every step has a profound purpose. This process ensures that life continues from one generation to the next, while also introducing the beautiful variety that makes the natural world so rich and dynamic. Understanding meiosis is like understanding the secret recipe that life uses to create both stability and endless novelty. It is truly one of the most beautiful and important processes in all of biology.